Good evening. This is Srinivas sir. You are watching Untold Botany. In this session, I am going to discuss plasma membrane, otherwise cell membrane, and the same is called plasma lemma. All are one and same. What is the plasma membrane? This is the outermost layer of the animal cell. Because in animal cell, there is no cell wall. So, presence of the cell wall is a unique feature of the plants. You know very well. As cell wall is absent, plasma membrane itself, it is the outermost layer of the animal cell. Whereas in the case of plant cell, outermost layer is a cell wall. So, interior to cell wall, plasma membrane is present. So, plasma membrane is present in between the protoplasm and cell wall in the case of plant cell. Okay, you see the cell membrane, the term cell membrane was coined by Nageli and Kramer. See, Nageli and Kramer are the workers who coined the term cell membrane. For the same, J.K. Plover coined the term plasma lemma. So, plasma lemma was proposed by J.K. Plover, whereas the cell membrane was coined by Nageli and Kramer. However, all the plasma membrane, cell membrane, plasma lemma, all are one and same. So, that is the outermost layer of the animal cell and the innermost layer of the plant cell. Here, if you see the plasma membrane is selectively permeable. What is the selectively permeable nature? See, according to the permeability, the membranes are of various types. Like, I think you are very familiar with all of them. Permeable membrane, impermeable membrane, semi-permeable membrane, selectively permeable or differentially permeable. So, there are 4 types of membranes. Permeable membrane means which allows both the solutes as well as solvents through it is called permeable membrane. Which allows neither the solutes nor the solvents is said to be impermeable membrane. It does not allow the solutes, even the solvents. Whereas in the case of a semi-permeable membrane, it allows all the solvents but does not allow the solutes. So, the membrane that allows the solvents and does not allow the solutes is said to be semi-permeable. Then what is this selectively permeable? The new one. Selectively permeable nature means it allows all the solvents. In addition to that, it allows few selected solutes. So, that is, here selectively permeable membrane means not only it allows the solvents, but also it allows the few selected solutes. Such membrane which allow all the solvents and few selected solutes is said to be selectively permeable membrane, and the same is called as differentially permeable membrane. Here our plasma membrane is selectively permeable, otherwise semi-permeable. That is nature. So, coming to the position, just now I revealed where it is present in the plant cell, innermost. Innermost means outermost. What is that? Cell wall. So, outside the plasma membrane, cell wall. Inside the plasma membrane, protoplasm. So, plasma membrane is present in between protoplasm and cell wall in the case of plant cell. Whereas in animal cell, plasma membrane is present in outside the protoplasm, exterior to the protoplasm, that is the outermost layer of the animal cell. Okay, coming to the size of the plasma membrane. Plasma membrane, it consists of 75 angstrom units, nearly the size. And the composition, what it is made up of, what are the chemical components present in the plasma membrane. I think all of you are very familiar with that. Plasma membrane is made up of lipids, proteins, even carbohydrates. Here, if you see the lipids you take, so which type of lipids are present in the plasma membrane? Mostly phosphoglycerides, phospholipids like lecithin, cephalin, such ones are present. Lipid molecules are amphipathic in nature. What is that, Nana? Amphipathic. Amphipathic nature means the same molecule exhibits both the hydrophobic nature as well as hydrophilic nature. If you see the lipid molecules in the plasma membrane, lipid molecules are arranged in a bilipid layer. Means two monolipid layers together constitute bilipid layer. You can see this. So it is a monolipid layer, lower one. Upper one, another monolipid layer. Two monolipid layers together constitute bilipid layer. So always lipid molecules in the plasma membrane are arranged in a bilipid layer. Two monolipid layers together constitute a bilipid layer. In that bilipid layer, if you see the a molecule of the lipid, it consists of head. So this is the head, and whereas the other side is, and this is tail. So this is a tail, this is the head. So head is hydrophilic, water loving nature. Hydrophilic, whereas the tail is hydrophobic. So tails are hydrophobic, whereas the heads are hydrophilic. So same molecule exhibiting the two properties, hydrophilic nature, even hydrophobic nature. Such molecules which exhibit both the hydrophilic and hydrophobic natures are called as this. So what are they? Amphipathic molecules, and the nature is called amphipathic nature. The same molecule, it exhibits both the hydrophilic nature and hydrophobic nature. So which is hydrophilic and which is hydrophobic? Head is hydrophilic and tail is hydrophobic. Hydrophilic heads are oriented outer side. You can see in this bilipid layer, these are the heads. So these are the heads. Heads are oriented towards outer side, whereas the tails are oriented towards inner side. So tails of both the monolipid layers come together here. Means they are oriented inner side, whereas the heads are oriented outer side. So this arrangement of the lipid molecules ensures that the hydrophobic tails, which are made up of that saturated hydrocarbons, are prevented from the aqueous environment. So to prevent them from the aqueous environment, tails are oriented towards inner side, whereas the heads are oriented towards outer side. So here, in between the tails of the monolipid layers, van der Waals forces are present. Because of them, they are held together. And in between the heads of the lipid molecules and outside the lipid molecules, proteins are there. In between the proteins and lipids, their ionic bonds and hydrogen bonds are present. So in between the proteins and heads of the lipid molecules, ionic and hydrogen bonds, and in between the tails of the monolipid layers, van der Waals forces. So due to this, a bilipid layer is formed. It is present. Always the lipids are present in the bilipid layer. So amphipathic, as I said, it exhibits both the hydrophilic nature as well as hydrophobic nature. For instance, lecithin, cephalin, such lipid molecules, mostly phospholipids. So that is a type of component. What is that? Lipids. So coming to the one another, what is that? Protein. So proteins that are present in the plasma membrane are of different types. According to the position of the proteins, there are two types. What are they? Peripheral proteins. Second is integral proteins. The names themselves they indicate whether what are they. Peripheral proteins. Peripheral proteins means the protein molecules that are present on the bilipid layer loosely. You can see here, it's a peripheral protein which is present loosely on the bilipid layer. For instance, spectrin. Spectrin is a peripheral protein which is present and on the surface of the bilipid layer and loosely attached on it. Whereas the integral proteins are bound tightly in between the bilipid layer. You see, this is a bilipid layer, in between them tightly bound. So the proteins which are bound tightly in between the bilipid layer are called as integral proteins, and whereas the proteins which are loosely associated on the surface of the bilipid layer are called peripheral proteins. Peripheral proteins are also called extrinsic proteins, whereas the integral proteins are also called intrinsic proteins. For example, the transport proteins, they come under intrinsic or integral proteins. Whereas the peripheral proteins, as I said, spectrin, cytochromes, they come under peripheral. These two types are depending on the position of them. Coming to the function of them, again proteins are of three types, like structural proteins, carrier proteins, catalytic proteins. Structural proteins which make up backbone of the plasma membrane. Carrier proteins which involve in the transportation, like porins. Many other proteins are there. Carrier proteins which carry out the facilitated diffusion. And coming to the catalytic proteins, catalytic proteins are nothing but enzymes like ATPase. So proteins present in the plasma membrane are of three types on the basis of their function, such as structural proteins, carrier proteins and catalytic proteins. Based on the position, two types. What are they? Peripheral proteins, integral proteins. Like this, different types of proteins are present and lipid molecules also present. One another component is carbohydrates. Yes. Actually, the structure of the plasma membrane was unknown until the invention of that electron microscope. After invention of that, this was revealed, particularly based on the studies on erythrocytes, in which proteins are of 52% and lipids are of 40%, and 2 to 3% carbohydrates also present in the plasma membrane. And mostly carbohydrates are present in the form of oligosaccharides. So what are the oligosaccharides? Here I have written oligosaccharides. What are the oligosaccharides? These are the small chains of the monosaccharides. Small chains of monosaccharides are said to be oligosaccharides. So carbohydrates are oligosaccharides. Some now and then they are covalently connected with the either proteins or lipids. Their association with the lipids results in the formation of glycolipids, and their association with the proteins results in the formation of glycoproteins. So glycoproteins, glycolipids are also present. They are derivative compounds. So like this, in the composition we come across through proteins, lipids, and carbohydrates in the form of oligosaccharides and glycolipids and glycoproteins. All are present. So this is the composition of the plasma membrane. Okay, how these components of plasma membrane are organized in that membrane. So what it is the arrangement? To explain the arrangement of these lipids and proteins and carbohydrates in the plasma membrane, various workers proposed various models. Of them, some are rejected and some are accepted. So let us have a brief account on the models. Coming to the first model, trilamellar model. So trilamellar model. 3 layers present, hence trilamellar. Proposed by Davison and Daniel Lee. Davison and Daniel Lee are the workers who proposed trilamellar model. According to this model, there are 3 layers in the plasma membrane. 2 are dark colored layers like this, and 1 is light colored layer like this. So these dark colored layers represent protein layers, whereas this light colored layer represents a bilipid layer. So 2 monolipid layers together constitute bilipid layer. Hence 2 protein layers, 1 bilipid layer. Together 3 layers. That is a trilamellar model. And the same is called sandwich model. Sandwich, an edible item, which is made up of 2 bread pieces and in between them some stuff is present. The bread pieces are equal to the protein layers, and whatever the vegetable stuff present in between that bread pieces is equal to the bilipid layer. So it can be compared with the sandwich structurally. Hence it is also called sandwich model. As 3 layers are present, it is called trilamellar model. It was proposed by Davison and Daniel Lee. And the same model is called PLLP model. PLLP means, so this is protein, this is monolipid layer, again this is monolipid layer, again this is protein layer. So protein layer, lipid layer, again protein layer. PLLP. So PLLP model is nothing but sandwich model, otherwise trilamellar model, that was proposed by Davison and Daniel Lee. According to this model, the thickness is 75 angstroms. Protein layers constitute, each protein layer constitute 20 angstroms. 2 protein layers together constitute 20 plus 20, 40 angstrom units, and the lipid layer constitutes 35 angstrom units. Together it constitutes 75 angstrom units. And in between the lipid layers here, in between the tails of the lipid molecules, van der Waals forces, and in between the proteins and heads of the lipid molecules, hydrogen bonds and ionic bonds are present. So this is a model proposed by Davison and Daniel Lee. And here one another, Robertson is a worker who proposed a unit membrane model. This is not a new model. This is a new name to the trilamellar model. Actually, this person, Robertson, he came across through all the biological membranes. Many biological membranes are there in the living systems, not only the cell membrane. You take tonoplast, membrane of the vacuole. Like that, many membrane bound cell organelles are there, either unit single membrane bound or a double membrane bound, like chloroplast, mitochondria, or lysosomes, vacuole. You take any cell organelle membrane, in all the biological membranes, nuclear membrane you take, any membrane you take, in all the biological membranes, he came across through the same organization of the components of the plasma membrane. All they show similar organization. Hence, he changed the name to the unit membrane model. Unit membrane model was proposed by Robertson. Actually, all these models are rejected because they failed to explain the selective permeability of the plasma membrane or elasticity of the plasma membrane. They could not explain the selective permeability of the plasma membrane and the elasticity of the plasma membrane. Hence, later, one another model, it was popularized. That is nothing but fluid mosaic model proposed by Singer-Nicholson. Singer-Nicholson, he proposed fluid mosaic model. According to Singer-Nicholson's fluid mosaic model, here proteins are present in the form of icebergs in the ocean of the lipids. So, in the ocean, some here and there, icebergs are present. Like that, in the ocean of the lipids, some here and there, protein icebergs are present, like gulab jamun in the concentrated solution. That can be compared. So, this fluid mosaic model, you see, this is a fluid mosaic model. This is the bilipid layer. These are the heads. These are tails. Again, here heads. So, they are the oligosaccharides. And these are the proteins. Various proteins like peripheral proteins and integral proteins are present now. So, this protein is a peripheral protein which is loosely associated on the surface of the bilipid layer. You take this protein, it traverses throughout the length of the plasma membrane. Such proteins are called as tunnel proteins. Tunnel proteins. And this protein you take, this protein, half is outside, half is inside. So, it enters into the plasma membrane. Such proteins are called transmembrane proteins, like ATPase, F0, F1 particles or oxysomes. And ATPase enzyme, which will be discussed in the photosynthesis, here it consists of F0, F1 particle. F0 is embedded within the membrane, whereas the F1 is projected outside the membrane. So, such proteins are called as transmembrane proteins. If they traverse throughout the length of the plasma membrane, so they are called as tunnel proteins. Even the porins also come under the same transmembrane proteins. So, porins are the proteins which are useful in the transport. If they transport water, they are called aquaporins. These porins are present in the bacteria and the mitochondrial outer membrane and the chloroplast outer membrane. In them, we come across through the porins. How the fluidity is caused in the plasma membrane? The fluidity is because of the phospholipids. Because most of the phospholipids are made up of unsaturated fatty acids which are liquid in nature. So, because of the presence of these unsaturated fatty acids, the plasma membrane is fluid nature. Actually, this is quasi-fluid. Quasi-fluid means semi-fluid. This fluidity or fluid nature is because of that unsaturated fatty acids which are present in the phospholipids. So, phospholipids cause fluid nature to the plasma membrane. Like this Singer-Nicholson's fluid mosaic model is widely accepted, whereas this trilamellar model and that unit membrane model proposed by that workers were rejected because they could not explain that elasticity, even the selective permeability of the plasma membrane.